Okay, welcome to this week's Biological Sciences Seminar. So before I talk about Dr. McCravy, I want to say, so next Wednesday, we will have a former alumni, a guy named Joe DeBold. He's working for the Missouri Department of Natural Resources, so has done lots of work with things like peregrine falcons,
Lots of deer,
deer,
elk, all that kind of stuff that is happening in Missouri.
Yeah.
And he's a very dynamic speaker. He's going to talk about opportunities. Probably half the dead animals in the mammalogy lab were accredited to him. And so he's a really fun speaker to have.
Yeah, okay, so that's next week. So today's speaker is Dr. Ken McCravy. He is here from the University of Georgia. I know Ken, and many of the faculty know Ken here, because he worked here at WIU for 20 years where he was an entomologist. During that time, he was my best friend and I think 85% of my social life. So I'm really happy to have him back. It's been a long time. So he retired about COVID time, I think, and so has been down in Georgia since then. Ken, or Dr. McCravy, is originally from Atlanta, Georgia. He got his undergrad at the University of Georgia, which is in Athens, where REM and the B-52s are from, and so super hip at least once in history. He got his master's degree from Old Dominion, which is in Virginia. Then he got his PhD. He returned to the University of Georgia. So while he was here at WIU, that was about 20 years, he did work in Illinois on the effect of habitat variation on insect diversity. And I think some of the things he'll talk about today are similar to that kind of work. While he was here, he also spent some time in Honduras, where he studied diversity of orchid bees, which are important pollinators for things like vanilla. Is that right? I'm making this stuff up. That's not on my list. So yeah, I'm pretty sure vanilla is an orchid. So okay. I do want to say that Ken was a super steady and productive scientist while he worked here. I could always find him at his desk. He published close to 35 articles on insects while he was here, and he mentored over 20 students. So a lot of master students, a lot of honor students. Now, I'm pretty sure he is enjoying retirement. He is working as a research professional at the University of Georgia in the Department of Entomology. So Ken is here to talk today about beneficial insects of North Georgia, apple and peach orchards. Okay, yeah.
Thanks, Sean, and thanks everybody for inviting me back. As Sean mentioned, it's been a few years, and I'm glad to be back here and the opportunity to tell you about some of the research that I've been involved with since I moved down to Athens to the University of Georgia. When I
think. Try it
now. Oh, okay. Yes, when I moved back to Athens, as far as Zoom is concerned.
That vicinity, yeah.
Oh, okay. So when I moved back to Athens, I was interested in continuing in research, at least to some extent. And so I went and talked to some professors at the university in my old department, and met Dr. Brett Blau here. And Brett is a associate professor in the entomology department, and his position is primarily extension and research. And he works with insects, mostly pest insects of apple and peach orchards. But he also is very interested in more, basic biodiversity of these orchards, which is what I've been working with since I joined his lab. And when I first joined the lab, my main assignment was to help him out, or help out a couple of his graduate students who were sort of bogged down in their research and their processing samples and so forth. One of these students, Jordan Bailey, was a master's student who had been collecting samples for her master's in North Georgia apple and peach orchards. But she was doing double duty as lab manager. So she and sort of, excuse me, fallen behind in processing her samples. So that was my main assignment there, was to help her out with her samples. But in the process of doing that, I became very interested in a group of insects that she was collecting in her samples, the hoverflies. And I started trying to identify her hoverflies to species level, which if anyone's tried to identify insects to species, it's pretty challenging, especially a diverse group like the hoverflies. So it's very slow going. Fortunately, I was in luck because in the department we had a postdoc, Dr. Scott Clem, in another lab, who was a hoverfly expert. So lucked out on that one. I approached Scott and asked him if he would like to help us out with our hoverfly identifications, and maybe we could get a publication out of it. And he was on board with that. And then we also brought on Sabrina here, who's a PhD student of Brett's, and a good statistician, good background in statistics. So she helped us out with our statistical analyses. So this was our hoverfly team here. Okay, I'll tell you just a little bit about hoverflies. Most people, when they think of flies, they think of pests, house flies and mosquitoes and black flies that transmit diseases. But a lot of fly groups are very beneficial, and the syrphids are one of those. They're a very diverse group, about 6,700 species, almost 300 genera, and they're sometimes referred to as dual ecosystem service providers. That is, many hoverflies are pollinators. They visit flowers, and hoverflies are generally considered, after the bees, to be the most important group of pollinating insects. Many hoverflies are also predaceous in their larval stages. And in particular, they feed on aphids, which are very serious pests of agricultural crops. Aphids, they have piercing, sucking mouth parts. They suck out plant juices, and in the process can transmit some pretty serious plant diseases. So hoverflies do us a real service by feeding on those aphids. Okay, another kind of interesting thing about hoverflies is that a lot of them are pretty good mimics of Hymenoptera, bees and wasps. And of course, they gain some protection from that by mimicking insects that can sting. And so here's a couple of species that we collected in our studies. The most common of which was Toxomerus geminatus here, which looks like a little wasp. And over here, Aristolis resembles a honeybee. So, and then we see here, this is the larval stage of Toxomerus feeding on an aphid. So, demonstrating their pest management activities there. Okay, so the question we were asking with Jordan's syrphid flies is, are there spatial and temporal patterns of hoverfly diversity in North Georgia peach and apple orchards? And her study was done up in North Georgia, well, down in North Georgia, and at a couple of sites, a couple of orchards, one near the town of Blue Ridge, Georgia, and then a second one a little bit south of that, near Ellijay. Here's Atlanta and Athens for reference. But in each of these two sites, well, here's a couple of shots of the sites. These are peach trees that are in bloom. And then here's kind of an overview of one of the orchards. That's a malaise trap. You can ignore that. We didn't use those. And that was another study. But at each site, we back up a little bit. A lot of orchards in North Georgia, apples and peaches are grown basically side by side. So it makes for a nice comparison. So at each of the sites, we had a peach orchard and an adjacent apple orchard. And so in each orchard, peach and apple, we set up bowl traps. And bowl traps are sometimes called bee bowls, or just little colored bowls with preservative or soapy water, something to preserve the insects that get trapped in them. Often or usually different colored bowl traps are used because there's a lot of evidence that different insect species are attracted to different colors. So typically yellow, blue, and white bowls will be used to get a diversity of insects in the sample. So again, very good for bees, very good for hoverflies, as well as other insect groups. So in each orchard, we set up 8, or Jordan set up 8 of these 3 bowl arrays, 4 along the edge of the orchard, and then 4 interior, about 25 meters interior in the orchard. And so did that in both the peach and the apple orchards. And surrounding the orchards on this side, at both sides, was basically undisturbed vegetation, forest. Okay. And then so over here we'd have more peach orchards, and over here more apple orchards. So our edge traps were intended to help us see if there was maybe some hoverflies moving from the forest bordering these orchards into the orchard, see if there's an edge effect. Okay, and so then we identified our insects and so forth, and used generalized linear mixed models to compare the hoverfly assemblages that we found in apple versus peach orchards, and then also bloom versus post bloom. As I mentioned, these peach and apple trees bloom pretty early. They actually bloom in late winter down in Georgia. So peach trees a little bit earlier, late February, early March for a couple of weeks. Apple trees early to late March. So we wanted to see if, or get an idea, was there more hoverfly activity during bloom than immediately after bloom? Right. So that, and then we wanted to again compare edge versus interior to see if maybe there's movement of the hoverflies from the nearby vegetation into the orchards. I should mention that backing up here, the apple versus peach. Apple trees are obligately insect pollinated, okay? Whereas peach trees are not. They can be wind pollinated. Insect pollination, there's evidence that it helps, that it may improve development of the fruits and so forth, but peaches are not obligately pollinated by insects. Also in apple orchards, there are pest species that aren't present so much in peach orchards. In particular, some aphid species. The green apple aphid is a big pest of apple orchards in Georgia. So those are a couple of differences between apple and peach trees that led us to predict that we might find more hoverflies and greater diversity of hoverflies in the apple orchards than in the peach orchards. Okay, so here's our results, our species that we collected. We collected 15 species of hoverflies, and you see a couple of them were really dominant here: Toxomerus geminatus and Toxomerus marginatus, two very closely related species that accounted for over 80% of all the hoverflies we collected. Another Toxomerus down here, was one of the more common species, as well as, Xylata ajuncida. These three species of Toxomerus are all flower visitors, so potentially pollinators, and they're all, aphid predators, so potentially they can provide, pest control. This other species, Xylata ajuncida, is sort of a, unusual in its habits. It, feeds on pollen, but usually what it does is land on leaves and feed on the pollen that's on the leaf surface. So they're not really important in pollination. And their larval stages are saprophytic, so they feed on decaying organic matter. So Xylata ajuncida probably not real important in pollination or pest control. But these other three species of Toxomerus could be. Okay, so how did our hoverfly abundance and diversity compare in terms of crop species, apple versus peach, bloom period, bloom versus post-bloom, and edge versus interior? So we looked at three different parameters: hoverfly presence, were there hoverflies in the traps, hoverfly species richness, so how many species did we have, then hoverfly diversity using the Shannon diversity index, so a combination of richness plus relative abundance of the species. And what we found in terms of the crop species, pretty clear pattern, at least in two of our three measures here, of greater hoverfly presence and richness in the apple orchards compared to the peach orchards. In terms of hoverfly diversity, there was a hint of a pattern here, but not quite statistically significant. In terms of bloom versus post-bloom period, the collected or greater hoverfly presence post-bloom, as well as species richness post-bloom. So after bloom was when the hoverflies were most abundant and had highest species richness. Or, excuse me, this was not quite significantly different. So these two were significantly, there was a significant difference between bloom and post-bloom in terms of richness and diversity of hoverflies. Then not much going on in terms of edge versus interior habitats. So when we add a fourth comparison, Toxomerus abundance, remember Toxomerus was by far the most common genus we collected in sampling. When we look at Toxomerus abundance in terms of crop species, bloom period, and habitat, abundance greater in apple orchards, okay, so which, consistent with what we found with overall hoverfly diversity. But when we look at Toxomerus abundance in relation to bloom versus post-bloom, we get an opposite pattern. Lots of Toxomerus during the bloom period. So, potentially that might suggest that the Toxomerus are visiting flowers and maybe doing some contributing to pollination. Then again, when we look at edge versus interior, Toxomerus abundance, no significant difference there. Okay, so to summarize this part of the study, or this part of the talk, hoverflies, more abundant, diverse in apple than in peach orchards overall. And overall, more hoverflies during post-bloom, but Toxomerus showed the opposite trend, more Toxomerus during the bloom period. No differences in edge versus interior habitats. So, this, I think, it's interesting that, again, that the hoverflies were more dominant in the apple orchards again. They may be providing pollination services and pest management services. It was a little bit of a surprise to us that we didn't get any differences in edge versus interior habitats. There have been studies done in mostly in Europe that have found pretty strong patterns of greater hoverfly presence and abundance in the edge habitat compared to the interior habitat. What may be going on is in most of those studies, the surrounding environment is very fragmented forest. Okay? So in our study, basically these orchards are kind of in the middle of a sea of pretty much undisturbed forest. So it could be that there's so many hoverflies in the surrounding landscape that you're just sort of, inundating the orchards, and so kind of, eliminates any difference between the edge and interior. There's as much hoverfly, presence in the interior of the orchards as in the edge. At least that's one possibility. So definitely some, more studies that we can follow up with on this. Need more information on the role of these hoverflies in pollination and pest management. We're kind of guessing that maybe they're important, but we don't really know how or if the hoverflies are contributing very much to pollination or pest management. Some more studies of that, landscape effects on the hoverfly fauna of these orchards, and also effects of orchard management on hoverflies. These orchards are subjected to pretty intense insecticide treatments, herbicide treatments, pruning, and and so forth. So they're pretty intensively managed. And so, what effect is that having on the hoverflies? I would imagine, especially the insecticides, are having a pretty serious effect on the hoverflies. But again, maybe it's a short-lived effect. Maybe there's hoverflies in the surrounding landscape that can recolonize these orchards pretty quickly. But we don't really know that. Okay, so that's what we've been doing with our hoverflies. As I mentioned, I've been involved in a couple of studies of students of Brett's. So the second one I'll describe to you was done by Daniel O'Connell here, and he's another one of Brett's graduate students, a master's student who kind of similar to Jordan, got bogged down in processing his sample, so I helped him out. And his study involved effects of ground cover management on ground-dwelling arthropods in peach orchards. And again, John mentioned, I've done some work with ground beetles here at Western, so had some familiarity with ground beetles, so I started trying to identify the ground beetles, but discovered that I'd lost my touch in the last few years. Fortunately, luck was with me again, and I occasionally do work at the Georgia Museum of Natural History. And one day I was down there kind of rearranging insects and so forth, and I met Kurt Harden here, now Dr. Kurt Harden. At the time, he was a grad student at Clemson, a PhD student. And you guessed it, his specialty is ground beetles. So I approached him, similarly to Scott, and asked him if he'd be interested in taking a look at our ground beetles and helping us with species identifications, and maybe we could write a paper on it. And the answer was affirmative again. So, and then we had again statistical expertise, this time from Dr. Connor Fair, who's a statistician with the University of Georgia. So it's kind of his job to help with stats. Okay, so ground beetles and tiger beetles. The ground beetles and tiger beetles, up until a few years ago, were all included in the family Carabidae. Since then, the tiger beetles have been separated out into their own family, the Cicindelidae, but still very closely related species. They're both in the superfamily Caraboidea. Okay. Very diverse group, particularly the carabids. Combined, we're talking roughly 40,000 species, so there's lots of ground beetles out there. Most of these are Carabidae, ground beetles. A couple thousand, 2,000 to 3,000 are tiger beetles. So a couple of very diverse groups, and also potentially very beneficial. They're, for the most part, generalist predators. They'll eat anything that doesn't eat them first. And they may, again, provide pest management services by feeding on pest insects with life stages that they're ground dwelling. In particular, tiger beetle are very voracious predators. Right? The ground beetles are, again, generally.
ไหนเนี่ยของคราว่าเดนเจีย
Don't know who that is. Please mute your
ได้ไหมหนูมา
phone.
apply multitraction
Last year, I mean, I applied it almost 2 years ago.
อ่า
2 years.
Yeah, I do. I
2022,
have
when I
seen-
applied it.
โอ้บามียังก็ไม่ทราบ apply อยู่ว่าเฮาเนี่ย
Okay. They're muted. I think it was now. I think it was Rebecca. Okay. Go ahead, Ken.
Yeah, so some of the carabids, the ground beetles, are actually seed predators. So they provide weed control. So, and then also, ground beetles in particular are often used as indicators of environmental conditions. So they're important in, assessing environmental damage and so forth. Oh.
Now I think we need to click this thing again.
Oh,
Let's see. Okay, try now.
cool. Okay, so our question with Daniel's data, how do ground beetle how do ground cover management practices affect ground and tiger beetle diversity and species composition? Okay, so Daniel's study was done over a 2-year period. He tracked for 3 months, May, June, July, 2020 and 2021, I believe. And so what his study involved, it was done at a peach orchard near the UGA campus at the UGA horticulture farm. And he had 3 ground cover management treatments: herbicide, mowed, and unmanaged. So this aerial view of the site here, you can see the herbicide treatments pretty clearly, just bare soil there. Kind of hard to tell the difference between the mowed and unmanaged from this view, but the mowed plots were basically kept mowed to a height of about 3 inches. The unmanaged plots, as the name would suggest, were just let go. Okay, so generally, overall, herbicide treated plots, little or no vegetation. The mowed plots, vegetation, but mostly monoculture grass. And then the unmanaged also, contain flowering vegetation and so forth. So his study was done in a randomized complete block design, 4 replicates of each of these 3 treatments, so 12 plots total. And each plot consisted or had a pair of pitfall traps, okay, to collect the ground and tiger beetles. Okay, then our stats for this project: indicator species analysis, right, which is pretty commonly used analysis to test for associations of particular species with particular habitats. In this case, for a particular ground or tiger beetle species, they're associated with certain ground cover management practices. Okay. Effective number of species is basically a measure of species diversity that sort of combine, back up a little bit, the different species diversity indices place different weights on species abundance. So for instance, species richness puts no weight on relative species abundance. Different species count the same whether there's only one individual or 1,000, right? Then other indices, for instance, the Shannon index, places some weight on relative abundance of species. Then the Simpson's index places even more weight on relative abundance. So it really emphasizes the most abundant species. So what we want to do with the effective number of species is take a look at species diversity at all three of those different levels. And and this was analysis that Cotter suggested and did. I was not familiar with it at all. Distance-based redundancy analysis, which is sort of a multivariate version of analysis of variance that we use to compare species, overall species composition in the different ground cover management plots. Okay, so our overall results, we collected, 18 species here, roughly, 350 or so ground and tiger beetles. Had 2 indicator species, Harpalus pennsylvanicus, which I showed you earlier, which is seed-eating ground beetle, was most common in the unmanaged plots and was a significant indicator species of the unmanaged plots. So over 90% of Harpalus were collected in unmanaged plots. Then down here to Tetraka, Carolina, okay, which I also showed you earlier, the tiger beetle, was the most dominant species overall in terms of numbers and was very strongly associated with the herbicide treated plots. So like almost 80% of Tetraka were collected in herbicide treated plots. Oh, maybe I didn't show you earlier. I thought I'd show this. Yeah, okay, so here they are. Now I'll show them to you. Here's Harpalus, kind of a medium-sized ground beetle, pretty nondescript black coloration. And here's Tetraka, pretty typical ground beetle, good size, almost an inch long, long legs, large mandibles, large eyes. So you can look at these insects and pretty much tell that they're sort of open ground, very visual, fast running predators. So not really surprising in either case that we find Harpalus in the untreated, primarily in the unmanaged plots, which is where you're going to have the most diverse seeds available. Okay, then also not too surprising that Tetraka here was most abundant in the herbicide plots with open ground, and because they can utilize their fast running and visual skills to catch prey. And should mention with Tetraka, they're a known predator of the brown marmorated stink bug. You're familiar with that? You probably may not know it, but they're the ones that get in your house in the fall. Yeah, they're really irritating. But they're also a very serious agricultural pest and the pests of orchards. And they have immature stages that crawl along the ground to disperse. And so with Tetraka being a predator of those, it's kind of interesting that they would be so abundant in the herbicide treated plots. Maybe they're preying on the stink bug larvae or immatures in those plots. Okay, in terms of effective numbers of species, again, this is equivalent to species richness. Q equals 0, that refers to the fact that there's 0 emphasis placed on relative abundance of species. It's just simply species richness. Q equals 1, equivalent to the Shannon index. Q equals 2, equivalent to the Simpson's index. You see, no difference in species richness, lots of variation. That's pretty typical with species richness analyses, because with species richness, again, a species counts as present, whether it's just 1 or 1,000 of them. So with species richness, you can get like vagrant species, or only 1 or 2 individuals of a species that may not be typically found in that habitat, but increase the species richness. So that produces a lot of variation in species richness, and so which means no real difference in species richness among the 3 treatments. But when we look at Shannon's and Simpson's index, the herbicide treatments were significantly lower than the mowed and unmanaged treatments. And that was really due to the dominance of Tetraka, the ground beetle, in the herbicide treatments, which when you have a dominant species like that, then it decreases the overall diversity measure. Okay, our analyses of the overall differences in species composition among the 3 ground cover treatments or redundancy analysis. And what we've done with redundancy analysis is kind of decrease the number of variables that explain these the species composition in these 3 treatments, and see if there's any statistical difference in those. And there was, in particular, you can see here, highly significant difference, but mostly due to the herbicide treatments, which the distance indicates that they're quite different from the other 2 treatments. And again, that's undoubtedly due to the dominance of Tetraka in the herbicide treatment plots, which makes the herbicide species composition very different from the other 2 treatments. Okay, so to conclude this part of the talk, again, Tetraka carolina, tiger beetle, and Harpalus pennsylvanicus, dominant in herbicide and unmanaged plots, respectively. And again, that makes sense when you consider the biology of those species. Herbicide plots, the lowest ground and tiger beetle diversity, and also the most distinctive ground and tiger beetle communities. Again, due pretty much totally to the dominance of Tetraka in the herbicide plots. One thing that I was a little surprised about with this study is we got such clear differences despite our small plot sizes. I don't think I mentioned, I should have, that these plots were like 4 by 8 feet in size, very pretty small plots, and I would have expected that the plots that small, you would have a lot of spillover, among the different plots, and that would sort of wipe out any potential significant differences between the treatments, but it did not. So makes one think that if you apply these treatments on an orchard-wide basis, then you really get an effect. And so in orchard management practices, should definitely consider their effects on the ground and tiger beetle fauna. For instance, and it's, kind of goes against the grain for an ecologist to suggest that maybe herbicide treatments would be effective in controlling insect pests in these orchards, because it would promote the abundance of tetraca, which is a pretty important predator, again, a round worm-rated stink bug, and undoubtedly anything else that crawls along the ground that it can catch and eat. Yeah, so wanted to mention a few other projects that I'm involved in the lab. Brett had a third student, master's student, who's since finished up, who collected data using pitfall traps on insects in commercially managed orchards versus organic orchards. And by commercially managed, I mean, lots of insecticides, lots of herbicides, very, chemically oriented management. So I'd like to take a look at the ground and tiger beetles in these in the commercially managed versus organic orchards and see if there's a difference there. Also, we got more studies going on up in in North Georgia, apple and peach orchards. We want to expand our studies into the surrounding landscape to see if we can detect some sort of effect of the surrounding landscape on insects in the orchards. Another study I'm involved in is more of a classical pest management study, I suppose. It's a survey of a couple of serious pests of apple and peach orchards, the oriental fruit moth and the codling moth. And so we just started this. We put out pheromone traps, again, in North Georgia, apple and peach orchards, put out pheromone traps for these moths. And we want to see if there's a difference in orchard type. When you read the literature on these moths, it lists apples and peaches as these insects being pests of those fruits, but it really doesn't give any information about do these insects prefer one or the other. So we're hoping to get at that question and also get an idea of the seasonal phenology of populations of these moth species. So that one's, we're just getting started with that. And then I mentioned I've been doing some work at the Museum of Natural History, and Scott Clem, the hoverfly guy I mentioned earlier, when he was at Georgia, he went through the entire hoverfly collection of the museum and identified everything, the species, which is a kind of a rare thing for insect collections. Most insect collections, a lot, if not most of the specimens will be unidentified specimens. Now we've got thousands of hoverflies that are identified as species, and this is potentially a really great source of information. What I'm doing is going back behind him and digitizing those specimens, which basically means entering the species, the collection information, locality, date of collection, and so forth into a database. And hopefully one of these years, when I finish that, then we'll be able to analyze those data and write a paper on the hoverflies of Georgia that summarize distributions in the state of Georgia where more sampling is needed, and were there species present decades ago that are no longer found in particular locations, things like that. Okay, I want to acknowledge a couple of undergrad students, Haley and Robert here, who helped us out a lot with both field and lab work. Mercier and Hardy orchards, the two orchards, that we've been working at in North Georgia. And, UGA Horticulture, Research Farm for allowing Daniel to work on their, site. And, again, we've been doing a lot of work with the Georgia Museum of Natural History. Rick Hobeck is the collection manager there, and he's been real helpful with finding specimens and facilitating our work and yeah. Okay, so references and yep. Got it. Well, we have time for.
Yes, yes, so thank you, Ken. Dr. Hum has class at 1. So,
Oh.
yeah, we have a little bit of time before she boots us out. So, yeah, so any questions for Dr. McCravy?
So with the hoverflies, when you collect them in your bowl traps, you said an important stage as far as being predators is their larval stage. But is finding the bowl traps an indication that they've set up and that they're emerging right there from, eclosing, or does it mean that they're potentially just coming in and then will later?
Yeah, that's a good point. We're not really sure. If that necessarily means, the larvae are found when we're collecting. So I would expect that.
I guess you could say, why would they be there if they weren't gonna lay some eggs, but.
So can I follow that up? So, are these hoverflies the only things that land in those bee bowls?
No, you get bees, other, flies, occasional wasps.
So then why did you choose to study hoverflies if there's other insects in there?
They just caught my attention. I thought they kind of cool.
I thought you were helping someone out. Are they super abundant or super important or?
Pretty abundant, and yeah, they're, considered potentially beneficial, and, I was just sorting through Jordan's samples and became really interested in the hoverflies, and mostly when I was sorting through her samples, I was just like identifying things to family. I don't have the expertise to go to the species on, so the hoverflies were a group that I was able to find a good online key and seeing like I could potentially identify species.
So you looked at do you remember, are syrphid flies the most abundant thing that was in the bowls, or is it
No,
mostly?
no.
Yeah, yeah,
Actually, a group of flies, the Lycopodidae,
no,
common names for long-legged flies, and they're actually another group we're really interested in. They're predators in the adult stage, and prey on aphids, so they could be important in pest control. And we caught I think a couple of thousand of those, and I've got a Lycopodidae taxonomist up in Montana who's agreed to identify our Lycopodidae. Actually has identified our first year samples. So yeah, hopefully we can get all those identified and then get a nice study from that. Yeah.
thanks.
And I'm curious about the flowers on the peach tree. You said that they were not obligate insects, pollination, but they are colorful, right? And presumably the pollen's relatively big. Can you talk a little bit more about that?
I wish I could. Yeah, they look like they should be attractive to insects. Don't they? Yeah, I'm not, unless, I don't know, maybe like in the selective breeding process, they lost their attractiveness. And I don't know. Yes, it's really kind of weird. You expect flowers that look like that when being during the insect pollinated.
Yeah, I'll look more into that. Thank you.
Maybe time.
So I know you mentioned both of these groups provide pest control. And so I was wondering, like, are there any programs where they try to use those solely as pest control as opposed to pesticides, or are they just not as effective?
I don't know if there are any, I don't know of any specific programs that rely on them. There are, programs where, like, surrounding vegetation is left undisturbed to promote ground beetle abundance or hoverfly abundance. So in the, like, site management process, oftentimes they take these groups into account when they're managing vegetation. But I don't know of any like hardcore biocontrol programs that focus on these foods. I think probably one thing holding that back would be rearing them in enough numbers, where you could release a whole bunch of them. So,
Okay, I think with that, we should stop. So thank you very much. And I'll say one last time. So the department and Dr. Musser are providing dinner at 6 at Los Toros. Please tell him if you
Or
If you plan to come.
Just get a head count, basically.
thanks a lot for driving so far.